As we setting his computer, I would like to introduce Professor Tuan Vo Dinh. He is the director of the Fitzpatrick Institute for Photonics at Duke University in North Carolina. And, well, many of you already know him from his books, right? He has many interesting books on light, tissue interaction, and everything. He has a great contribution in biomedical engineering, especially in the deep understanding of light tissue interaction. So even though we almost all know you from many ways, I guess now we're going to see you presently presenting a nice topic on plasmonic nanosensors and nanoprobes. So thank you, and you have the 30 minutes for go.
Thank you very much for the introduction. First, I would like to thank the Dr. Karachi and the conference committee for the kind invitation to give kind of overview of our work for the last, I would say, 10 years. First, I would like to tell why we want to detect early detection, what the cause. And then I give some introduction to plasmonics. For some of you working in that field, it's very simple, but for many of you not familiar with the plasmonic, I give some really quick introduction. And then I will select here 5 technologies that have been developed in my laboratory to kind of illustrate what you can harness the light from plasmonics for detection and also treatment of disease, and finding a quick conclusion. So biomarker detection, this is, I think, a paradigm shift in medicine. In fact, when people look at the future medicine, what we want to do is early detection, because it's the key of disease control. If you detect early enough, cancer, infectious disease, you really don't have to, treat, and especially the time and the cost. So what are the biomarkers that are very effective for early disease? So when you look at the stream, the kind of key chain, the first one is the change in the DNA, because that's on the top of the screen. So you have gene mutation, gene expression before you get to protein, before you get to that people feel sick. So that is the key. So when you do early enough, then of course the improve treatment. So this is where you look at biomarker in the early detection. Then before even the symptom appears. So I show some example detect at the RNA, mRNA level that people know before they know they get sick. So that is the key. So the other clinical demand, of course, is a practical way to detect early, precise, rapid, all the things. And also, the word that we recently, heard about is theranostic. If we can develop a seamless diagnostic treatment at early disease, then it is very useful. Well, for plasmonics, so what is a plasmonics for those of you not familiar? It's an enhanced magnetic field. So when you have nanoparticles of metals, let's say silver or gold, around certain size, I would say 30, 150 nanometers, and there's a lot of theory in that, and you shine light onto that. It could be light from sunlight, it could be light from a laser. Then, around the nanoparticles, there's the electromagnetic field of the laser make the conduction electron oscillates. It's crazy. And those oscillate create a strong amplified field around the nanoparticles, and that is a plasmonic. So it's that field. So that field, can enhance many signals. For example, the Raman signal that we talk later, about 10,000 times when you shine light, you have a Raman, which is scattered back. And the Raman is usually very weak, but when you have enhanced to the 3rd, to the 8th, then you have a very strong. And then, people have found that silver and gold, but now there's a more, let's say, material that create those strong field. And when you have, and I see some so-called hotspot, you can increase the signal to 10 to the 13 or 10 to 15 times. So, there's a lot of theory, there's a lot of calculation, I show some of that, but just for those of you, just in application, just remember this picture, this kind of, it's like a so-called lightning rod. Think about lightning rod, when you have a sharp spike, it's a concentrated electron field and it creates a strong field at the tip. So just remember, that is kind of a pictorial summary of plasmonics. So first technology, which is we call molecular sentinel. In fact, we invent that name for what? So what we use is the surface enhanced Raman enhancement. We depend on the distance of the metal nanoparticle and Raman. You remember that when I mentioned to you that the field is very close to nanoparticles, and we try to explore. And that's our first paper when we discover the effect. So what we do is that we create a stem loop. The stem loop has several parts. When the DNA, when nothing happened, it makes a loop here, and then the loop is designed to recognize the DNA, mRNA, a nucleic acid target, and the stem form stem loop. Here we have a thiol, and we put here a dye, a label, a Raman label. What happened when we have 0 nanoparticles, around 30, 40, 50, then the thiol kind of binds to that. So when it binds to that and we shine laser, as I mentioned before, the dye is close to the nanoparticles, it enhances the field, plasmonic enhancement. You have a strong signal. And you see it's very sharp. That's why we use Raman. Raman is sharp. It's kind of a signature of the molecule. When fluorescence is strong, but fluorescence is very broad. Suppose you look at the wavelength here, fluorescence will go like this, where the Raman is sharp, like a mass spec. So now what happened, if we have a target DNA in the samples, and you remember that this is designed to recognize this, so what happened is it wrap around, it hybridize, and when it hybridizes, it opens the loop. And when they open the loop, remember, the signal, because it's far away from the nanoparticle, it decreases. So you have a decrease. So it's designed to recognize the probe and decrease. So we have a couple of applications. One is, let's say, for breast cancer. We show that we can do multiplex. So you see when you have two probe, one is ERB2, which belongs to the GF receptor. And when there's nothing happen, it makes the loop. It has that peak. And we put a Sci-Fi 3 dye, which, and the probe belongs to the recognized ERB2. Then we have another probe, K67, which recognizes the K67, which is a cell population marker, and we use another probe. You see the red color, another probe. And these are two examples. So what happened when you have nothing in the samples? You have two probes. Those have two very sharp peaks from the black and the red dye. When you add two probes, when it recognizes two, ERB2, it opens both. The signal decreases. When you have one probe only, the target, only one open, and you see the 1, 2, 3 decrease only. When you have the other probe, the K67, you see only the 1', 2' decrease. So just an example to show that with SIRS you can do really multiplex. One thing which is very interesting I want to mention is that our technology, this one, is homogeneous assay. That means you don't need to clean sample. Hybridizing assay is very well known for 30, 50 years. But every time people have to clean the samples to remove non-hybridized. So you need microfluidics. Also, you cannot do in vivo assay because you cannot go to in vivo something that is non-homogeneous because you cannot clean. Here, you don't need to clean. Just it's open and closed and you see it. So the molecular standard technique has potential for label-free because you don't have to label the target and multiplex detection biomarkers. This is an example which is interesting for infectious disease. So we work with a group at Duke, Dr. Ginsberg, and they discovered that, mRNA called RSD2 is a mostly expressed gene in a host signature. So his group, in fact, we work with Univ technology, we have a DARPA grant, and each group showed that, he can detect people before they know they are sick. The idea is that, can you detect, people get fever, they get some symptom, but really to find them they're sick before they know it. So he discovered that one, maybe a dozen, and we work just as an exam on the RSD2. And they, in fact, the work is very long. In fact, they incubate healthy adult volunteer, and they look at, all these, microarray, and, use a mathematical job. So we try this with, okay. So we look at RST2 gene. We show that with no gene in there, very sharp. But when we have a gene in the samples, in blood sample, suddenly it decreases. So we were able to detect, and also we showed that we can quantify. So the idea is early detection using those nanoprobes. So we can look at infectious disease and using that. So now we'll look at the second technology, and it's based on plasmonic coupling. So before I talked to you, plasmonic, strong around, the particle. But if you have multiple particles, or you have a structure that between those gaps, when it's small, it's an even stronger structure enhancement. So these are so-called plasmonic coupling. So the field is very strong, and the enhancement could be 10 to the 15 times. And so what we want to do is that to use an assay based on the plasmonic coupling to detect, and how we do it. So we design a probe, and the probe has a linker, and I show the two nanoparticles. One has a reporter that that has a Raman dye, and one is a capture probe that will capture, recognize any nucleic acid. And notice that this is shorter than this, okay? So what happened, and you look at the thiol here, the 2-iol. So what happened is when nothing happened, they hybridize, they get to here. And in the sample without target, they hybridize. And when they hybridize, the dye in the middle experience the coupling, and a very strong signal. But now if you have in the samples, and you have a target, the target will come and hybridize and block the hybridization, so there's no coupling. So that is, we use here the reduction in the source density for, interfere with plasmonic coupling for detection. So a couple of examples. So we want to show that here that we can detect even a single nucleotide polymorphism, just a change of one nucleotide, the SNP. So we use HERB2, which is also one in the breast cancer gene. And, we use a PCI. And we design the probe with the Y type and with the SNP. And you see, this is a capture probe, this is a reporter probe, and then the Y type, nothing happened. But in the SNP, it's recognized with capture, and suddenly the signal decreased. So we show that then, the PCI can be applied for target label-free SNP detection. So here we look at microRNA. MicroRNA is a hot thing now, because microRNA is a kind of short, non-coding RNA, and people have found in a lot of disease. In fact, it found in tumor, it found in breast cancer, in brain tumor, in infectious disease. It is very powerful technology. So we were able to see whether we can detect, microRNA. So we test MIR21. MIR21 is in prostate cancer, in breast cancer, and we can try that. And so it overexpresses in a variety of cancer. So we developed a probe. And as you see here, if we have a probe with MIR21, it blocked hybridization. It blocked this, the reporter, and suddenly we have no signal. So this is also show that the PCI can be applied for target-free, label-free MIR detection. One thing we are excited is that when it hybridized and blocked hybridization, the color changed. So when there's no target, in the solution look gray, and when there's target, it starts yellowish because the aggregation. So what we are developing now for global health, a very simple, inexpensive diagnostic. So our dream, in fact, we are working with a group that tries to develop something for lymphoma in Africa. The idea is to go to a village in Africa, in the Amazon, where there's no clinics. You just take the blood sample, kind of drop it into a vial, shake it, look at the color change. And if you have some connection to the cloud, take the cell phone, take a picture, send to a cell phone, and it's sent to a clinic. So that's what, what we like very much is that here we use a very sophisticated technology that involves plasmonic, it involves a quantum mechanical kind of concept, but make it very simple. And just a color chain that can detect really, selectivity, a type of microRNA. So let me talk now about technology 3, which we call inverse molecular sentinel. So you recall with the molecular sentinel, we see the signal on first and it decreases. And then now we want to look at off to on. And the reason is that we happen to talk to clinicians that work with us. And they say, hey, Tuan, can you make it something that black when nothing happened, and you light up when things happen, and say, why you want that? Well, in the clinic, we like to see that, okay? And so we design. So before that, let me introduce to another nanoparticles that also we develop. It's a nanostars. And you recall that I mentioned to you, remember the lining rod? When you have a sphere, you don't have a sharp spike. But when you have a really, very nanostar, you have multiple, spike. So we have worked with some of you as well on nanorod. It has spike. But here, the nanorod is one spike, and you have to orient the polarization of laser light. Here you have multiple nanorods, so it's from the star. And we do calculations here, and we show, as you see here, at the tip of those nanostar, you have very strong electromagnetic field. One thing we're exciting is that we can design the the number of branches. And by changing the branch, we can shift into the near infrared. Because we want to do also like in vivo contrast agent for imaging and therapy. And you see here, we can move from, S5, which is a 5-star branch, to S10, we get into 800 nanometers. So that's also something that we can do with a nanostar. So what we do is that we design a probe with the gold nanostar. The probe has a Raman label, okay? The thiol here, bind to this. And then we design a placeholder. The placeholder is designed to hold straight. So the dye is far away from the nanostar, so you don't see a signal. So nothing, the SIR signal is off without the target, okay? Okay. So what happens if we put the target in? The target goes this, it starts to, we design the toe hole here, it binds to this, and then after it binds, there's a so-called a branch migration, it displaces slowly, and then when it's placed completely, it removes the placeholder, it's out, and suddenly this is free. And when this is free, it forms a loop. And the stem loop starts to have a signal. When you shine light, it suddenly is on. So before it's off, now it's on. So we tried with a couple of things. ISD2, I mentioned to you that, it's infectious disease. We put a label, we design the probe, and as you see here, when there's no target, a blank, when we put a non-complementary as a control, nothing happened. When the target ISD2 in there, you have a signal label. And we can quantify as well. So this just to show that, we can do some semi-quantification. And here, show you that we can detect 200 attomole with the current system because it's very sensitive. We will just put into a small capillary on the spot and can detect, the system. We show also multiplication with the breast cancer gene, the MIR21 and MIR microRNA-21 and microRNA-34A. And as you see, when there's a blank, there's no signal. When you put the MIR21, this will come up. When the MIR34, this come up. When we put two MIR21, MIR34, we have all this signal come up. So we show that really it can be multiplexed. So now let me talk now to technology 4. And the goal of technology 4 is to develop nanosensor for single cell monitoring. And the question is that, why do we want to detect single cells? Is that more simple to collect, thousands of cells, lyse them, freeze them, we have more signal? Well, single cell, when you look at the mechanic, if you want to understand what is going on with disease, it's very complex. This is a single cell of the fruit fly. And at any time, there's a 20,000 interaction between 7,000 proteins. So if you want to know something, we need nanotechnology. We need something that gets into the cell. So nanoparticle, I mentioned to you, is one of them. We developed a nanoprobe. So what is this? We pull the fiber down to 40 nanometers. And at the tip, the 40 nanometers, we can put antibody. I don't have the time to show some of the results of the slide here. We can go into the single cells of a tumor cells, and we can detect, let's say, biomarker of DNA. We use that nanoprobe to look at PDT. In fact, in a single cell, we expose the single cell to ALA, and then we shine laser, and we were able to get the single cell and show the life appearance of caspase chain reaction. So apoptosis in a single cell treated with PDT. So I do not have time to go that. I'd like to concentrate on the disease. So here what we do is we put at the tip of the nanoparticles, nanosensors, which is the very tip here, a dye. And this dye can change, the peak, just a very small change, when the pH change. So SIRS is very selective. Only the polarity can change the dye. And you see here, the peak change. And by looking at the peak of this dye here, we can calibrate from 6, go to 8. So what we do is that we work with one of my collaborators, Dr. Seawall. She's here. And, she's developed a technology by using a very tiny level to make aspiration of a very tiny amount of cells from women. And she has a theory about half dozen parameters to analyze the risk for breast cancer. And one of them is pH. So I've been collaborating with her. I do, my student postdoc come to her clinic. She take a few cells. We walk to our lab, which is about 10 minutes, and we put into our nanoprobe, the nanoprobe, the cell nanoprobe, and we can detect pH. And this is data of the depiction from, a clinical application of nanotechnology to show the pH. So this is the technology number 4 to go single cell analysis. So now let me go to the so-called technology number 5, which is, we talk about Go Nanostar, and here we want to detect not only diagnostic, but also therapy. And we try to work on a couple of tough problems. One of the problems we work is the brain tumor, the blood-brain barrier. The blood-brain barrier is incredible. So we have a discussion, at the yesterday session about what is a good size of nanoparticles. And as you see, the nanoparticles, when there's a less than 10 nanometer, they got filtered through the kidney and get into the urine. But when they are larger 100 nanometers, they get stuck by the liver cells. So only a certain, between 10 and 100, which is the main thing, that can remain in the circulation of the bloodstream long time enough, which you want. And then the blood-brain barrier, another thing that when you have tumor cells, they are leaky. If you can modulate around the 30, 50, they can accumulate more in the tumor cell. But the brain tumor is another story because it's very tight. It doesn't have that kind of opening. So it's very tight junction. So it's very hard. That's why, it's hard to cure brain disease because, the drug never gets into the brain. It protects our brain, but also it protects things from getting. So what we do here, and I hope that is work. Here, what we do, yes, it's work. So here, we discover also that the gold nanoparticles have a very strong 2-photon luminescence. And what you just see here is, the single nanoparticles. that we can inject into the window. So we have the mouse here. At Duke, we work with several people, Dr. Jerry Grant, who is now at Stanford. And so here is a window. We inject through the tail vein, and it goes to the tumor, and we can see where those nanoparticles live. And we see single nanoparticles in the tumor. So we go on the brain. So before it was a flank. Now on the brain, we have the window on the cranial of the mouse, and we can see incredible resolution, unprecedented spatial resolution through the cranial window on the live animal. And we notice that with normal area, you see the structures, the tumor has more, distorted, geography, I would say, than the thing. And I want to show you here, again, a very high resolution. So here, too back, the light is strong, but you can the 3D resolution of, of all the vessel at unprecedented resolution. So we were able to use nanoparticles. We can also, now another thing is that by shining lightly with light on those nanoparticles, on the tumor area, we can create a blood-brain barrier disruption. So we propose a new concept as a spatially controlled photothermal-induced blood-brain barrier disruption. So for example, you inject the drug to patient, you know where roughly the tumor area, you just put the light, and you create tiny aperture at the area, spatially controlled, which is very hard to look at to introduce the drug. So lastly, we want to show that those nanostar, we developed something multiple modality. This is an example, quintuple modality. Now we have set modality. We can do surface and Raman. We put MRI. Gold is very strong on CT, so strong concentration. We can detect by 2 photon luminescence, and we can do photothermal therapy. So this is a cartoon of the things. I don't have time to go in detail, but we can do CT because the contrast Asian, it can see sarcoma in the thing. Now I want to show you an example of SIRS, so alive. So you see when we inject, this is the normal area, there's no accumulation, but we control the size, the gold nanostar accumulate only on the tumor side. And if we use a Raman probe, we see that the SIRS signal on the tumor side, no signal on the healthy side. And this is something we show that we do. And we shine light here. This is a control, and you see we inject by the tail. It accumulates only on the tumor, So the tumor, the gold nanostar go to the tumor. Here, we inject PBS, not before treatment, nothing. After treatment with laser, nothing happened, the tumor grow. Here, when you have a nanostar injection, before treatment, it starts to grow. After treatment, it's gone. And we can see by CT that the tumor is completely gone. So here we show with the infrared camera to see that when we shine light, we heat this area very much. So the time is off now, so I just want to conclude that we can use Plasmonic to look at early biomarkers for disease, from DNA, mRNA, microRNA. And also those nanoprobes open new horizons for many applications, from diagnostic and so for therapy, we call theranostic. And very quickly, the acknowledgement of people in my group here. A lot of collaborators from the Duke Medical School. In fact, most of them are MDs. And the funding agency that help us. And also very 5-second about our institute, the Fitzpatrick Institute for Photonics at Duke, which is this building here. We have now associated with 110 faculty on 34 departments. And as you see, all the departments here, there's here are the faculties, and here are the multiple departments. It's not only from chemistry, physics, BME, but also for anesthesia, and you see comprehensive cancer center, vaccine, computer science, math, pathology, surgery. And with that, thank you for your attention.
So thank you very much, Professor Vo Dinh, for the nice presentation, a lot of new perspective in plasmonics and things. We have time for 1 or 2 questions, so if you have Okay. One there, one here.
Thank you very much for giving the very informative talk. What about the biocompatibility of these nano stars, especially of the gold and silver?
Excuse me, can you repeat again the question? What is?
Biocompatibility of the nanostars.
Oh, what happened of the nanostar insect?
Biocompatibility
Yes.
of the nanostar.
This is an incredible, great, important questions. So people working on that field look at there's two parameters, or multiple, but the two important is the type of the materials. So you notice when we use silver, when we do in vitro, because it's strongly enhanced at the sun wavelength, but when we go to in vivo, we use gold. Gold is biocompatible. In fact, at Duke, there's a professor looking at similar, different technology, but they go now to phase 2 clinical using gold nanoshell. So gold nanoshell has been for companies working. Gold has been used for arthritis for hundreds of years. So gold is one good material. Silica, people coat material with silica. Another factor which is important is the shapes of the molecule. So those are great questions, but a lot of interest and a lot of research going on.
Thanks for the talk, it was very nice. How about the size of these nanostars? Can you control it? And the one that you use for in vivo experiment, how large are they?
Yes, indeed. We can control. In fact, we can. So the technologies that we use, in fact, we develop it. There's several people. In fact, the nanostar is getting traction. You see a lot of publication nanostar, but most of other group use a CTAP, which is a very toxic ligand to develop. We don't use, and we can control the size and the number of branch, and we can shift into the different wavelength, so that we can control. Yeah, that's why you see that we were very excited. I couldn't show you, but we inject into mouse, and it go only to the tumor brain. So we are working on, we have now 80 mice working now at Duke, that to look how we can inject into, selectively due to the size.
So thanks for the talk. What's the time lapse, the average time lapse between the nanoprobe design and the clinical use? And what do you think are the main limitation to decrease this time lapse for us to have those nice applications in a widespread use?
The time lapse between development and use?
Average.
Okay. So that's a great question. In fact, let me give a perspective because the question is very big. So in the lab, okay, now, in fact, we are sending our nanostar to many places, to Russia, to Cornell, to many places. We found a place a way to do only for 10 minutes to go nanostar. So there's a trick there, but we do that. To inject, for example, usually when we inject to animals, we wait 30 minutes to a couple hours, depending where we want to accumulate. Now, the bigger question you might ask is that, when can we get into the clinic? So there's now in clinic, not gold nanostar, but we are another gold nanoshells and gold nanoparticles. It's in phase 2. So hopefully we can see in the clinics. And I noticed that there's in Egypt, there's a trial now also, a clinical trial.